Hi, everyone. Good morning and welcome to our West Hour. That's webinars empowering science and technology. Um, this morning, we have one of our very own, Dr. Jason Coleman from Precision NanoSystems, who is the clinical application scientist lead. He's going to be addressing you, and I'll let Jason share a little bit more about his background before he starts the presentation. So, Jason, please take it over.
All right, thank you, Kim. I guess welcome everyone. I'm happy to be here, part of the West Hour, and presenting to you today. I guess a little background about me before we get started. Obviously, my name is Jason Coleman. I'm a clinical application scientist lead with Precision NanoSystems, and I've been with PNI, I guess, almost 3.5 years now. I had started as a field application scientist, and now my role is as a clinical application scientist. So what that means really is, as we've seen a lot of clients, have success at the early stage of their formulations and looking to scale up those formulations to the clinic and ultimately to commercialization. I'm there as a resource to help with that on the technical side of things. So that kind of ties into what I'm going to be talking about today, with the title Scale Up with Manufacturing of Self-Amplifying RNA-LNPs for a COVID-19 Vaccine. So if you're wondering, and I'll get into this, what is PNI doing in this field? And it's really, you just see we have a lot of expertise and so as well as our technology, the expertise, to be doing some of this development ourselves. And ultimately, what I'll share with you today is that story, and really want to use that as a resource, as a learning tool for our clients, because a lot of our clients that they've had this early success and are looking to scale those formulations are really, we want to be a resource to help in that area. And I in particular want to be a resource to help in that area too. So I kind of hinted at that here is why is PNI developing a COVID RNA-LNP program when they're by nature a technology company. So back in the end of 2020, PNI had received a grant from the Canadian Strategic Innovation Fund, we call SIF, to develop a self-amplifying RNA COVID vaccine. And the rationale for that was because PNI is based out of Vancouver, and so from the Canadian government Canada wanted to have the capability to do this manufacturing within their own country. So with that grant came the development of the RNA-LNP vaccine itself, and then also a grant to build a biomanufacturing facility in Vancouver as well. So that construction is ongoing now. So from the timeline of this project, starting in early 2021, was the role of identifying, doing the in vivo and in vitro work to identify lead candidate selections. And what you'll see as I go through the talk today, talk in terms of what we did is we took 2 different formulations all the way through scale up, and you'll see there's a lot of, and really what that is meant to do is tell the story. Because a lot of times when we're in research, we're looking at the early phase work and seeing some of those early stage successes and getting really good results. But just keep in mind, or the idea is to keep in mind, you're going to have to eventually be able to manufacture and scale up these formulations, and what is that going to look like? So I want to kind of give you that full picture here today. So ongoing work now was earlier 2020 was the initiation of the safety tox study. We've since done several GMP manufacturing runs with the lead formulation, and then later this year looking to get these formulations into the clinic for phase 1-2 clinical trials. So if you look at what it takes to make a LNP vaccine, and really where does PNI fit into all of this? So we kind of always talk in terms of like the pillars of being able to do this, or we call the genetic medicine toolkit. So we talk in terms, first we start with our disease target. So that's kind of explanatory in this case, where we're dealing with a COVID vaccine. So our disease target is to produce the COVID spike protein. So we know that's ultimately our target, what we want to do. But then you look at what are the pillars, what is the necessities to make an actual vaccine. So in this case, I'll show you there's obviously different types of mRNA that can be used to develop these vaccines. We chose self-amplifying RNA. The rationale for that is, you need less of a dose, which requires a smaller manufacturing footprint. But through this program, and we've partnered with with other companies to have this expertise as well. But through this process, we've been able to develop that expertise needed for developing the payload itself. The payload being the saRNA. So that's one part of our expertise. And then the other component necessary, as we all know, or a lot of us I know nowadays, is you can't just deliver RNA by itself. It needs to be encapsulated in some sort of a carrier vesicle. For this work, obviously using a lipid nanoparticle. So PNI has the capabilities and also the resources. We have our own lipid platform for research use, and then we also have our own library of proprietary lipids. These are proprietary ionizable lipids that can be taken to the clinic. So the work that I'm going to show you today uses one of our ionizable proprietary lipids to do. And then what most people probably know about in terms of PNI is the nanoassembler manufacturing platform that we possess. And really, so this different stages of this platform were used for its development work to go all the way from the bench all the way up to GMP manufacturing. And then, of course, last but not least, is the reason that so all these reasons obviously were why PNI was granted the funds to do this, but obviously we had to have the in-house expertise to make this all happen. So what I'm really doing today is going to be just relaying that story to you. So hopefully that will be educational and useful as you go through your own development programs. All right, so kind of building upon that, as we know, not all mRNA is created equal. Different companies are utilizing different types of mRNA, both unmodified and the base modified, which is the vaccines that we know from Moderna and BioNTech, Pfizer. But the work we had done utilizes self-amplifying RNA or saRNA, and the rationale there is that because it's self-replicating once inside cells, the dose needed is much lower. I guess as an example, Moderna dosed around 100 micrograms. Pfizer, I believe, is around 30. But with with our current vaccine, we're looking into be in the dose range of just a few micrograms or less. So just a little background here. I know this is probably old news in a lot of ways, but I just kind of want to talk about what goes into a lipid nanoparticle. So obviously there's typically always four components: the ionizable cationic lipid, the cholesterol helper lipid, and the pegylated lipid. So different functions that all of these have. But so as I briefly mentioned, the work that we had done utilized two different formulations, and we take them all the way through manufacturing just to build upon that story of things in vivo and vitro work isn't going to tell you the full story. You need to really look at the manufacturability of those formulations. And as I had mentioned, the ionizable cationic lipid component to this is a proprietary lipid from PNI that we had used. All right, so from the technology side of things, how are we doing all this? How are we doing the scale of all this work? And really what a lot of people know about, this is really where people know about PNI is from our technology. And what you see here, so we use a microfluidic based mixing technology where you have two inputs into these microfluidics, one being your aqueous phase with your RNA, and the other being your ethanol phase containing your particle excipients. So this microfluidic mixer that you see in the top left here, that architecture is maintained throughout our entire platform. And that's really the kind of the heart and soul that gives this technology the scalability, so that at the smaller scale from the Ignite and Spark, from all the way up to the Blaze and GMP and ultimately commercial system, you can get reproducible and the same particle characteristics that you get across the different scales. So what changes as you go through this work, obviously with the scales, is the rates at which these materials can go through the cartridge, and thus relates to that is the volume capacities that can be done. So the work that I'm going to talk about today, the initial work was done on the Ignite, and we did 0.2 milligram batch size, and then the Blaze instrument that you see in the middle here, that's where a lot of that process development work was done. So obviously when you talk about the whole process, the microfluidics, the part where the particles are formed, that's extremely important, but it is one part of the whole process. So when you're going from Ignite to Blaze, how you're doing that downstream purification is going to be different. Like for example, with Ignite, you can do things like Amicon centrifugal filtration or dialysis, but on the Blaze, you get into needing larger scale purifications where you get into things like tangential flow filtration. So really and what I'll talk about is that process of tangential flow filtration, all the variables that you need to look at and evaluate. And the Blaze is a research tool that gives you the capabilities to do that. So you can do it in a research setting with very minimal waste volumes, because we all, so I'm sure at this point, know and appreciate the cost of RNA, and in a lot of cases, lipid components too. So you can really, really figure out what all those variables are before you get to your final GMP manufacturing. And then the final phase of that is, so the Blaze, we did about 2.5 milligram scale. And at the GMP scale, so for the phase 1 studies, we need a batch size of 50 milligrams. And that, a lot of you may seem very small, but keep in mind with the self-amplifying RNA, that those dose requirements are very minimal. So, and we know from going from the Blaze GMP, it's an easily scalable, transferable process to do that, especially if you've already ironed out that downstream process with the Blaze. And then finally, so that's the instruments that were used for today. And then just as a note, I guess, so this is for clinical phases, but as you are looking to scale up for commercial grade, we do have a system that can be kind of customized or together to meet those needs as well. All right, so another kind of thing to note here, in the last year, we were purchased by Danaher, and then we've now have these sister companies, Aldevron, Paul, and Cytiva. And what that means is we now have the full capabilities to go all the way from RNA synthesis all the way up to the final fill and finish of the drug product. So really, the goal there is to be able to provide clients a true end-to-end solution for LNP manufacturing. So, but the one thing to know, like even though a lot of these systems are in place, so obviously here the big component of it that we focus on a lot of times is the particle assembly piece itself, which is where the nano assembler instrument platform comes into play. But a lot of the work when developing this process goes into the downstream part of that, which for the purification piece will be tangential flow filtration, and then finally sterile filtration, or then sterile filtration, and finally fill and finish. But the one of the reasons why it takes a lot of expertise to develop the downstream tangential flow filtration process is you got to remember a lot of these TFF systems were not designed or optimized with lipid nanoparticles in mind. A lot of them were used for cell banks and cell purification and things like that. So, it does take some work. It does take some knowledge to get these systems to be optimized. And then, the other thing to note here is that with our NanoAssembler instrument platform, if you're making LNPs at the small scale, you don't need expertise in order to do that. And you'll know that, those results are going to be transferable to larger scale systems. And something also new to this year that's worth noting out is that, a lot of, we do now work with CMOs and CMOs and want to provide our platform to enable their success. And then also with kind of the technology we've learned from this program, be able to help and ensure success from a just information standpoint as well. And then something else new this year is that there are no longer any technology access fees or royalties associated with any of our equipment. All right, so let's get into the study itself. So the goal here was really to take the self-amplifying RNA lipid nanoparticle using our own ionizable lipid, and then take that in the range from the bench scale, so 0.2 milligrams, all the way up to our clinical manufacturing scale of 50 milligrams, and then get that en route to a phase 1 trial. So a lot of the variables I'm going to get to or focus on is more going to be, as well as the initial kind of in vitro and vivo work, talk about the where a lot of the variables comes into play in developing this process, and what that is again is the TFF process. So a lot of these variables and I won't get into a ton of them in detail. In a lot of this case, I'll kind of just tell you what we use and just say these are things to look at. But it's going to be things like the type of system, a lot of variables around the membranes themselves, around the buffers, and then around the rates you're running the system at, and then of course the scalability, going from one scale TFF system to a larger scale. But in the end, what we want to do is, and the goal here is, which we ultimately did, was select a lead RNA-LNP formulation that meets a lot of this criteria in terms of still having activity, having good stability, being scalable, repeatable, and address a lot of those critical process parameters, and being able to have a good understanding of what those are, and then ultimately how they affect the critical quality attributes of our drug products. So again, here's the instruments that were used to do that. And we used the Ignite, Blaze, and GMP system. And as I mentioned, the Ignite was 0.2 milligram scale, Blaze 2.5, and GMP 50 milligram scale. The volume ranges of these instruments. With Ignite, you can go up to a 20 mil, but really for a 3-to-1 ratio, that's about 13.3 mils. With a Blaze, you can get up to 10 liters, and then with the GMP system, up to 50 liters. So well within the range of what we needed for this study. Flow rates, as I mentioned, as you scale, this microfluidic technology enables you to run at higher flow rates and get the same particle characteristics. So with the Ignite, these are the flow rates that we use for these studies too. The Ignite studies were done at 12 mils per minute, Blaze at 115, and then the GMP runs done at 200 mils per minute. And then those were all done at a pretty typical aqueous to lipid phase ratio of 3-to-1. And as I mentioned, at the Ignite, you're doing dialysis, and then on the Blaze and GMP systems when you're developing that TFF system or those TFF systems. All right, so here's just kind of a precursor to some of the results. So I mentioned, and you'll see kind of throughout this, we took 2 different formulations, LNP1 and LNP2. And without getting into a lot of detail, I can kind of note that LNP1 is a more fusogenic composition than LNP2. So what you may see from in vivo and vitro results, again, may not necessarily be as favorable under manufacturing conditions. So here you see, so LNP1, LNP2. So LNP1 is in, I guess that's a light blue, and LNP2 is that funky green color. But what you show here is that, so this is particle size versus PDI. And then so there's a graph on the left, and then percent encapsulation is on the right. So you see here from going from the Ignite to Blaze to GMP, particle size and is maintained, and then PDI, cutoff runs be below 0.2, that's all maintained. You can see, and in all cases, we get very good encapsulation efficiency. I think upwards of like 97, 98% was pretty consistent to what we got. So here's the first example. I'm not going to get into a lot of I'll mention it briefly at the end, but get into a lot of the, obviously a lot of analytical work goes into this, and there's a lot of analytical studies required for release testing, burst testing of your acceptance of material, in-process testing, and then testing for your final drug product release. But one kind of critical feature in all of this is going to be some sort of a bioassay, which ultimately measures the efficacy of your formulation. So in this case, the bioassay that we had used was adding the LNPs to HEK293 cells and then measuring the spike protein. So we can see, and what is good to see here is you can see a dose response, which is something that you want, because then you can ultimately be able to quantify the effectiveness of your drug product. So here's LNP. We'll get LNP1, LNP2. So again, two different formulations. You both have good efficacy, but you see the LNP1 does have slightly higher production of the spike protein. So then, as well as in vitro studies, we'd also done two different types of in vivo testing. First being basically giving mice the dose of the vaccine, both the prime and then a boost. So the boost was given 4 weeks after the prime, and then blood serum. So this was an intramuscular injection, and then blood serum was taken at 3 weeks and 6 weeks, and then the samples were measured for the IgG, and then specifically the neutral antibodies against the SARS-CoV. So what you see in this case, you have a naive, you have your just a GFP mRNA, and then you have your two formulations, LNP1 and LNP2. So you see both at 3 weeks and 6 weeks, you do get expression of IgG, but more importantly, you show both formulations, so neutralizing antibodies specific to the CARS or the COVID itself. And then the final animal study I'm going to go over briefly here is a hamster challenge study where the hamsters were ultimately vaccinated and then exposed to the COVID virus. Again, basically at 4 weeks apart was the dose, and after the 4 weeks apart was 4 weeks apart from the initial to the boost, and then 2 weeks after the hamsters were exposed to COVID, and then this study was done by measuring the weights of the hamsters and the weights of their lung to body ratio, collecting blood, looking at those neutralizing antibodies, and also the nasal terminate. So just this is apologies, this is a little busy, but just to go over kind of high level some of the results here. You can see here, so this is A is the control in black, and then B and C is going to be, in this case, will be the lead formulation with two different candidates of saRNA. So what you see here is that the hamsters that received the vaccine were able to maintain their body weight. They were able to not have a high increase of lung weight relative to body weight, meaning without the vaccine, the hamsters were getting a lot of inflammation in their lungs. And then here's the case on the right graph here. You see there's the IgG expression and neutralizing antibodies that you see with the vaccinated hamsters. And then the two graphs on the bottom here, looking at the viral load, both the control and then the two vaccinated groups. So what you see here is that doing a nasal wash in the nose area, you see much less of a viral load for the vaccinated hamsters. And then finally, with the tissue viral RNA study, the lungs themselves, and here different portions of the lungs were looked at, the cranial and caudal, and what you see is a much lower viral load with the hamsters that received the vaccine. All right, so now the fun part, at least for me, talking about the manufacturing itself. So just to kind of walk through what that manufacturing process looks like, right? So we have the microfluidic piece, which is where the LNPs are formed. So where the RNA gets encapsulated inside the lipid nanoparticle. And then as you know, we know once you make the LNP, if you're doing a 3-to-1 ratio, it's in about 25% ethanol. So from a stability standpoint, LNPs aren't going to be stable. So you need to lower that ethanol concentration, and you do that as part of the process, part of the nanocellular equipment, which is what we term here inline dilution. And then for stability, you think because some of the first formulation, LNP1, we knew potential stability issues based on the components. So we typically we try to get for dilution even as low as less than about a 3% ethanol concentration before going into TFF. So the next steps are TFF, where that's where you're going to remove your ethanol concentration even further, and then put the particles in what their final storage buffer is going to be, followed by sterile filtration, and finally fill and finish to get the drug product. All right, so in terms of TFF itself, right, I mentioned kind of the what it's used for, but we can see here there's a lot of different variables that you need to consider when developing your TFF process. Unfortunately, I guess there's not an off-the-shelf TFF system to use for lipid nanoparticles, because every formulation is going to behave a little bit differently. So you need to do this work to really understand your formulation going through the TFF process. So just some examples of variables that you need to look at, and I'll kind of just cut to the chase here and tell you what we had decided on for doing this work. But with the caution that I would suggest doing your own development work as well. So for the types of cartridges that you use, you can use hollow fibers or plates. We had most success, and we used hollow fibers. Materials, cartridges we use both polysulfone and modified polyethyl sulfone, so PS and NPS. Both will work. We had slightly better flux and better success with NPS. Molecular weight cutoff. For manufacturing, for R&D work, we tend to use potentially less than 100 kD cutoff, just because we want to really focus on particle retention. But for manufacturing standpoints, we typically use in the range of 100 to 500 kD. And a lot of times this work was done last year. So if anyone has done TFF, they know we're kind of the headache of being able to get TFF cartridges. So it's good to understand, what is a range of will and won't work. And that can sometimes be based on the availability of supply of some of those cartridges. So cartridge surface area, obviously this will change with scale. And then shear rate. So shear rate is ultimately you can basically say flow rate, but it's basically when you're exposing these particles to TFF and putting them through these fibers, you're going to be putting shear stresses on the particles themselves. And again, this is potential forces that the particles weren't necessarily intended to. So you want to be able to increase shear rate to increase the speed of your process, but not to the point of where it becomes possibly detrimental and the effects aren't really gained anymore. And then transmembrane pressure. So transmembrane pressure is really the pressure within the fibers, and it becomes a driving force for pushing material through the membranes. So again, here's on the right here is kind of another schematic of what that process is. So when we're after the dilution step with TFF, you'll do a concentration step initially, and the concentration step is really to as you concentrate your particles, you can first reduce the volume requirements of your TFF system, and then the volumes of some of those buffers that you need for the buffer exchange or diafiltration process. And then we had done, a series of different buffer exchanges to get into that final storage buffer, and then final concentration, which is determined what that encapsulated RNA amount is, and then again sterile filtration and then vialing. And for this case, we did requirement. We kind of looked at the WHO guidelines for what's needed for a pandemic setting, and in this case, we knew storing at -80 C was okay, but obviously understand the future, the benefits of not requiring cold storage or being able to do things like lyophilization. But for this particular product, the requirement was indeed -80. So here's just one example of one of the tests that was done for developing the TFF processing time. So there's a variable where shear rate was looked at. And again, 3 different shear rates used here were 4,000, 6,000, or 8,000 inverse seconds. And we can see again, I mentioned the process of TFF. So if we had the TFF coming off of the nanotumbler, and after dilution, you reconcentrate your particles. And then in this case, we did 2 different buffer exchanges. We basically found you need to use an intermediate buffer, because if you get right into your storage buffer, the flux rate was greatly reduced. So we did 2 different diafiltrations for volume exchanges. And then you see here, so going from 4,000 to 6,000, you saw a pretty significant reduction in processing time, and then not as much of a reduction going from 6,000 to 8,000. So it's not just, so in developing your TFF process, it's not just, does it work? Does my particle characteristics maintain? In all these cases, it is, but I think you're going to also scale up this process. So the amount of time that TFF is going to take has to be a factor that you need to consider. So this was done at the Blaze batches, a 2.5 milligram batch. And then here, just to cut the chase, we use a 300 KD molecular weight cutoff and a TMP of 4. But you got to think a lot of different testing was done even to get to this point, like similar to the testing done for the shear rate test. So here's just an example, and another kind of important thing to note when doing this process development. The more sampling you can get, the better, because we know that the particle characteristics are going to be maintained coming off of the microfluidics. So it's a lot of the steps in the downstream process where things can change, really, or the particle characteristics can change. So we want to look at take samples on different steps of the TFF process. So here's your concentration step, diafiltration, after concentration, and after filtration. So here's another example of LNP1 in the blue, and LNP2 in the greenish blue. And you can see throughout the different stages of the TFF process, the particle characteristics are fairly well maintained. Definitely within the acceptance criteria, but you do start to see a slight hit. Perhaps, I guess it's now I look at it a little biasedly, where you start to see a slight increase of particle size with LNP1 formulation versus LNP2. It's pretty solid throughout, but in both cases, and kind of throughout this whole study, your encapsulation efficiency is maintained again at that like 97 to 98% range. So this is a Blaze scale, 2.5 milligram scale. So then when you go up to the full clinical manufacturing scale, 50 milligram scale, what you see is that the particle size of LNP1 really increases. So it's really affected by those stresses at the larger. Because the TFF system, a smaller scale system was used at the 2.5 milligram scale, and then the larger scale TFF system was used at this scale. And so you see going through that process, the LNP1 formulation particle size greatly increased. But you can see here, so luckily, I guess, through this development, there's the take home message here is that there's, even in our case, there wasn't really a like an early stage study or an indicator necessarily to show that this would happen. So luckily, we had 2 different formulations that we were able to take to this level of development. So we had LNP2, which worked extremely well, even at this scale. You can see the particle size is maintained, and again, encapsulation efficiency is well maintained. But what this showed was that we knew LNP2 could withstand the downstream processing conditions. And again, and as with any kind of drug product you're developing, time of getting to your phase 1 study and ultimately getting to the market is critical. So with more time, I'm sure a TFF process could be developed for LNP1, but we had a formulation that worked. It had good in vivo, good in vitro activity. So we decided to move forward with LNP2. And that's now going through and take that through to GMP manufacturing and ultimately to the GLP toxicology studies. So what you see here, so this is an example now of the GMP manufacturing, so the full scale manufacturing. And what's kind of interesting here is you can see, so different sites, like totally different groups, totally, even different countries basically were able to duplicate this process and duplicate these particle characteristics. You can see here again, particle size being maintained, and then PDI being well within the acceptable limit as well. And obviously encapsulation efficiency is good throughout. And it's also good too to have this secondary check. We use cryo-TEM to just show the particles are very uniform and ultimately have that dense core and they look like they should. So it's nice to get that second verification. And then so another part of this is going to be looking at your stability. So the top graphs here are LNP1 and the bottom 2 graphs are LNP2. Graphs on the left are size, graphs on the right are PDI. So you kind of, if this was an indication, maybe this is an indication where with LNP1, you do see a trend of both increasing size and PDI versus LNP2, the size and PDI is pretty well maintained at different storage conditions. All right, and I don't get into a lot of detail here talking about, we could do a whole nother, obviously there's a whole nother webinar on just the analytical piece itself, but it's just worth noting, along with all this development and having come from industry before joining PNI and worked in the scale up and manufacturing and everything, I'd always been told, the analytical piece takes 10 times as long as doing the actual manufacturing. So just kind of have an appreciation for what it takes to develop those analytical methods. But just kind of know, here's kind of like, you want to be able to have the characterization of the LNP itself, and that includes size, PDI, lipid composition, and then the ratio of lipids to RNA. Then the RNA quality itself, and there's different ways to look at that. Basically the acceptance of the RNA and having a way to measure quality in that case, but also how do you measure quality once it's as part of your drug product, and that's where things like your bioassay come into play. Looking at encapsulation is important because we know that when we do a dose, it's based on the amount of encapsulated RNA, because that if the RNA is not encapsulated, it's not going to have any effect. And then obviously looking at more the payload itself and different kind of things you can do to the RNA, which is, that's a little bit beyond the scope here, but just things you can do of the RNA to make it more effective too. All right, so just in conclusion here to wrap up, so the take home messages, I guess. So we looked at 2 different lead formulations with LNP1, LNP2, and we're able to take those all the way to full scale, from bench all the way to full scale on the nano-assembler platform, the Ignite, Blaze, and GMP. And then we saw there at small scale, the Ignite and Blaze scale, the particles had comparable particle characteristics. And when I say the scale, I'm talking about the after downstream processing, because as we know, that's where the particle size was increased was a result of the downstream processing. But you saw too that the LNP2 or the LNP1 formulation at the larger scale did not handle the TFF process as well. And then finally, so we've made several 50 milligram batches at 3 different sites, just to show the robustness of the platform and also the process that we had developed. And we're currently made a GMP patch with LNP2 for QC release, and then that is being used for the GLP tox studies. So I see a lot of work was done by people other than me for doing this. So a big thank you to all of them. And I think hopefully I've learned a lot from this and will be able to help a lot of you that are going through this process. But I know as a group, we certainly have the collective knowledge to be a resource and provide the technical expertise as you are also going through this process. So with that, I'm done the talking, and I'll open up for questions.
No, thanks, Jason. It was great to hear. Really interesting to kind of see the pathway as it was followed.
Thanks, Ian, and thank you, Jason, for a very well-received, informative presentation today. Thanks to the audience for all their great questions. Just as a reminder, I wanted to introduce the team to you so you can reach out to us if you have any additional questions or want the presentation. My name's Kim Killian. I'm the Regional Sales Manager for the Southwest Territory, located in San Diego. My field application scientist is Viet Nguyen, who is located in Southern California as well. We comprise the Southwest team. Then, of course, our Northwest team is Angel Galarza, who's our director and who works very closely with Ian. Ian is actually located in the Pacific Northwest now. And we are happy to continue the conversation, introduce you to Jason if you wanted to have any further communication with him. And then we just wanted to remind you that all of our webinars are on our YouTube channel, and you can find that under West Hour, and that's w.e.s.t. Please look forward to our next Journal Club on April 27th at 9 a.m. Pacific time. And I guess that's everything for now. We'll see you soon, and have a great rest of your week. Thanks.
Thank you.